Environmental DNA, often abbreviated as eDNA, refers to the genetic material obtained directly from environmental samples, such as soil, water, or air. This genetic material comes from organisms present in the environment, and analyzing eDNA provides valuable information about the biodiversity of a particular ecosystem. The use of eDNA has revolutionized the field of environmental monitoring, offering a non-invasive and efficient method for studying the presence and diversity of species in various habitats. Here's how environmental DNA works and how it can be used to monitor biodiversity. 1. Collection of environmental samples. Environmental samples are collected from the habitat of interest. These samples can include water samples from rivers, lakes, or oceans, soil samples from terrestrial environments, or air samples from the atmosphere. 2. Extraction of DNA. The collected samples undergo a DNA extraction process to isolate genetic material. This extraction can be performed using various methods, and the resulting DNA may come from a variety of sources, such as cells, tissues, scales, feces, or other biological materials shed by organisms into the environment. 3. PCR amplification. Polymerase chain reaction, PCR, is often used to amplify specific DNA regions of interest. This step allows for the selective amplification of target DNA sequences, such as those specific to certain species or taxonomic groups. 4. DNA sequencing. The amplified DNA is then subjected to DNA sequencing, which determines the nucleotide sequence of the genetic material. DNA sequencing technologies provide information about the identities of the organisms present in the environmental sample. 5. Species identification. The obtained DNA sequences are compared to reference databases containing genetic information from known species. This allows researchers to identify the species present in the environmental sample based on genetic similarities. 6. Quantification of species abundance. In addition to identifying species, eDNA analysis can provide insights into the abundance or relative abundance of different species in a given habitat. The quantity of DNA from a particular species can be correlated with the population size or biomass of that species. 7. Non-invasive monitoring. One of the key advantages of eDNA monitoring is its non-invasive nature. Traditional methods of biodiversity monitoring, such as capturing and observing organisms directly, can be disruptive to ecosystems. eDNA analysis allows for the study of biodiversity without direct contact with the organisms. 8. Detection of rare or elusive species. eDNA analysis is particularly valuable for detecting rare, elusive, or cryptic species that are challenging to observe directly. The genetic material shed into the environment can be detected even when the organisms themselves are difficult to find. 9. Early detection of invasive species. eDNA is used for early detection of invasive species. The genetic traces left by invasive species in the environment can be identified before their physical presence becomes evident, allowing for timely intervention and management. 10. Monitoring changes over time. eDNA monitoring provides a dynamic view of biodiversity, allowing researchers to track changes in species composition over time. This is crucial for understanding the impact of environmental changes, climate change, or human activities on ecosystems. 11. Applications in conservation biology. eDNA has wide applications in conservation biology, aiding in the assessment of the effectiveness of conservation efforts, monitoring endangered species, and assessing the overall health of ecosystems. 12. Challenges and considerations. Despite its advantages, eDNA monitoring has challenges, including potential contamination, degradation of DNA in the environment, and the need for rigorous sampling protocols. Researchers continually work to address these challenges and refine eDNA methods. In summary, environmental DNA, eDNA, analysis is a powerful and non-invasive tool for monitoring biodiversity. By analyzing genetic material present in environmental samples, researchers can identify species, assess abundance, and track changes in ecosystems over time. eDNA monitoring has diverse applications in ecology, conservation, and environmental management.